In this chapter with the fun subject of DNA nanotechnology. So from the last module, you'll understand then that if you take a DNA molecule, double helical, with the bases specifically paired along here, and you make one side of it longer than the other, so let's just pretend the bases hanging out are these, and I won't draw 8 bases, I'll just draw a few of them, then a piece of DNA with a complementary sequence hanging out on another strand, so let's have a look. A would pair with T, T with A, C with G, and G with C, can come together to form a single double helix. So what you have is an addressable sticky end. Because this molecule will only form a double helix with its partner with a complementary sequence. That's one part of the story. 20 years ago or more, Ned Seaman realized something else. There is a process in the development of embryos, or the formation of embryos, called meiotic recombination, where, let's take mother's DNA here. So this is a double helix. And let's take father's DNA here. The sequences cross over and form double helices if they are homologous, that is, they have their complements on either strand. The purpose of this process is to mix up mother's and father's DNAs as part of a Darwinian strategy for making newly conceived organisms healthy. But what Ned Seaman realized was if you now make something like this a finite length, in biology these so-called holiday junctions will propagate long distances on DNA. If instead, and it's drawn out in this slide now with the strands shown like this, if instead you make it of a fixed length and you include overhangs so that pieces of it are specifically addressable, you now have an addressable corner element that can hold other structural elements in place. And what Ned Seaman realized, the actual three-dimensional structure of this is more like the corner of a box. It does something like this. And then with the third leg sticking out here, or the fourth leg. What Ned Seaman realized is now you had modules with which you could make any desired nanostructure. The only requirement was that you work out the right sequence of DNA, synthesize them, put them together, anneal them, which requires heating them up and cooling them down, in the presence of ions, typically divalent ions, because these lower the electrostatic repulsion between pair DNA helices better than single valent ions because of the e to the 2e term that appears in the exponential of the Debye theory. So, turning now to examples of DNA nanotechnology, this slide shows something called a double crossover. So it's two pairs of double helices crossing over. It's a little bit hard to see because it's put in dark blue here, but there is a common strand, and I'm following it along with the pointer on my mouse, that zips together all the other double helices in this junction. So here is what happens when you have tiles with sticky ends dissolved to associate with one another. They form a structure like this. And here's an atomic force microscope image. The distance between the corners of these tiles is 20 nanometers. And you can see that the predicted structure has a little hole in it right here where the 4 pairs of double helices bend away from each other. And you can see the little holes quite clearly in the AFM image. So the beauty of this technique is that you can make arbitrary nanostructures simply by designing the right DNA sequences and heating and cooling the mix. When you compare this to the problem of making nanostructures by top-down methods, for example, it's quite amazing. It has another advantage, and in my view, one even more important than the ones that I mentioned to you before. Let's take a double helix like this, and let's insert into it a piece like this. So this now sticks out of the nanostructure one is making, and it has some unique sequence here of A, T, C's, and G's. This is an address. It's a chemical address to which you can add anything, so this blob stands for anything. We attach to it a piece of DNA with the complementary sequence, and now it will be fixed at this point in the structure. We call this a molecular pegboard. Now, DNA technology has advanced yet further owing to a wonderful method developed by Paul Rothman, and I'm showing it to you here. This is called DNA Origami, after the Japanese word for paper folding. And it works the following way. Instead of taking a mixture of short strands and annealing them together, one takes one very long strand. In the example shown here, it's actually a circular genome from a virus, something called M13 phage. And this has around 7,000 bases. And so you say, how would I want to fold this to make a given nanostructure? Well, supposing part of the process is wanting to cross-link this piece to this piece. Of course, you want to introduce many cross-links for the final structure, but supposing that was one of the steps. One could take a small piece of DNA that formed a double helix here by base pairing, and then I'm showing a dashed line because the structure is not yet folded together, and then anneal with this over here to form a second double helix. So now the structure would look like this, where you have a little loop like this hybridizing across the structure. So in this slide, we show this schematically. So the goal here was to make a structure that looks something like the Michelin Man. So you see the black strand there is what would be the viral DNA. These little loops here are the staples that hold it together. And then every now and then, where there's no room for a staple because you've got a staple in the structure above, little pieces of DNA are filled in to make it double helical and rigid. This technique is so versatile and the yield so high that Paul Rothman was able to make this gorgeous structure that you see here. So here is how you might like to fold a viral DNA. No staple strands shown in this figure to keep it simple, to make a smiley face. And here is a nanoscale smiley face made by DNA Origami. So here's an example of the yield of DNA Origami. This is an AFM image of little origami tiles, one of which is blown up over here. In this case, it's a simple tribute to Arizona State University, ASU. But you can see that the yield of these tiles is almost 100%. Every tile is fully formed. The letters were made by hybridizing in loops. So if this is the background origami framework here, then at various points we put in loops like this, which themselves formed double helices. And so they look like little rigid blobs on the surface of the material. So we have made devices in which, so this might be about 50 nanometers, and this perhaps 100 nanometers, in which the pixels like this are used as a barcode for identifying a particular chip. And then over here are single strands of DNA that, when they hybridize their target, become rigid, so show up in the atomic force microscope as a high point on the device. The non-hybridized strands give no contrast. This illustrates the potential of this technology in biochemistry, biomedical analysis. Clearly, it also has potential as a way of prototyping nanostructures in many parts of chemistry, materials science, and engineering.